So we did peripheral chemoreceptors, we did central chemoreceptors, sweet deal. Now let's go ahead and talk about some of these actual receptors over here in the lungs. So let's first talk about, let's do stretch receptors. Let's do that one first. HerringBroiler Reflex. All right, so we're going to talk about the stretch receptors. So stretch receptors are going to be located in two different areas, okay? What color should we make these? Let's make these this doo-doo brown color. For some reason I like this color here, okay? So look, here's actually going to be some stretch receptors right here. Oh, it should not be right here. It should be in the visceral pleura, not the parietal pleura. So let's put this here. It should be here in the visceral pleura, not in the parietal pleura. And then we're also going to have some other stretch receptors, which are going to be located right here. Let's put another one right here too. Okay, so we're going to talk about these guys and what they're actually doing. Okay, so some of these stretch receptors are actually, one is actually located, some of them are located within this visceral pleural layer. So these stretch receptors can be located within the visceral pleura. And these other stretch receptors can actually be located within the smooth muscle of our bronchi and bronchioles, of bronchi and some of the actual smaller bronchi, which are called bronchioles. Okay, so whenever there's actually an increase in the ventilation, right? There's an increase in the ventilation process. You're inspiring, okay? And let's say that you're taking in a lot of air. I'm talking a lot. Let's say that's actually you're taking in air that's greater than 800 milliliters. So usually these are usually stimulated whenever the inspiration process, let's say that the tidal volume is actually, the amount of air that you're bringing in is actually greater than 800 milliliters. So the volume that you're bringing in, let's actually say volume here, volume of air that you're bringing in is greater than 800 milliliters. So you're taking in a decent amount of air. Whenever you take in this air, it stretches the lungs. Whenever it starts actually pulling the actual what? The visceral pleura closer to the parietal pleura and the chest wall is actually expanding, right? As the lungs are expanding, it reaches a maximum capacity whenever it goes to greater than 100 mils. And these guys, 800 mils. And these guys get stimulated. There's stretch receptors in the visceral pleura, as well as the stretch receptors which are located within the smooth muscle of the bronchi and the bronchioles. When these are stimulated, they're connected to a special nerve. This nerve is actually going to be called cranial nerve 10, vagus nerve. And man, this vagus nerve, he's something. So he receives these signals from these stretch receptors, and he sends these signals into two places. One is it can go in here and it can go to the pneumotaxic center, it can go to the apnoistic center, and it can also go to the DRG. Okay. So now, if this is bringing this information in, what is it going to do? Okay, well, it's going to tell the pneumotaxic center. And whenever it tells the pneumotaxic center and this apnoistic center, it's going to be like, okay, we're at the peak point of inspiration. So whenever you're at the peak point of inspiration, you're going to want to stimulate this guy. So this guy is going to be stimulated. And you're also going to let this guy know too, so you're also going to stimulate him. And then what these guys are going to do is once the pneumotaxic center and the apnoistic center are receiving the stimulus and the DRG is receiving the stimulus, they're going to send down, so this pneumotaxic center will inhibit the DRG. The apnoistic center, he will also provide inhibitory impulses to the DRG. Now look what else will happen. This actual information coming directly from the vagus nerve is also going to directly inhibit the DRG. So again, what happens? Let's explain it one more time. Stretch of the lungs, greater than 800 mils, activating the sensory afferent fibers of the vagus nerve, taking this information into the pneumotaxic center, the apnoistic center, and the DRG. What's the overall effect? The overall effect is the inhibition of the DRG. If the DRG is inhibited, what will it do to the VRG? It will inhibit the VRG. If the VRG is inhibited, then what's going to happen? The inspiratory neurons are not going to fire anymore. Then the expiratory neurons will. So again, let me explain this one more time. Information comes down the actual vagus nerve. We'll let the pneumotaxic center know, let the apnoistic center know, and let the DRG know. Overall result is inhibition of the DRG. When the DRG is inhibited, it will not send down any more inspiratory signals. Also, it will inhibit the inspiratory neurons of the VRG, but stimulate the expiratory neurons of the VRG, and then what will happen? Expiration will occur. Now, the frequency of these action potentials will be decreased, right? And if these action potentials are decreased, if the frequency is so low to where it's almost absent, right? Then what's going to happen to the actual impulses going to the lungs? It's going to decrease, or they actually become none. So really, we should say that it actually should stop. So the action potentials that's actually triggering inspiration should stop, and the action potentials that are going to trigger expiration should begin. And then what happens? What will happen to the lungs if actually inspiration stops? If our diaphragm doesn't contract, and if our external intercostals aren't contracting, what happens? The lungs go back down. They expire. What is this reflex called that actually triggers this action here? And it was caused by the stretch receptors within the visceral pleura and the smooth muscle of the bronchi and the bronchial smooth muscles. Whenever they're stimulated by volume greater than 800 mils, send information to the pons, to the DRG, which inhibits it, inhibits the VRG, and stimulates expiration. That whole reflex is called the HerringBroiler reflex. It's called the HerringBroiler reflex. Okay? Now, the HerringBroiler reflex is, again, that whole mechanism due to stretch receptors. Okay, so that should make sense. And again, the overall result is basically what? The overall result, stretch, send signals to the actual medullary respiratory centers and the pons respiratory centers, and do what? Inhibit inspiration and begin expiration. That's all it's designed to do. Inhibit inspiration, begin expiration, allow for the lungs to recoil. That's it. Okay, what about these guys? These guys look irritating. You know why? Because they're very irritant receptors. They're special irritant receptors right here. Okay, so here you're going to have your pseudostratified ciliated columnar epithelial tissue. That's your mucosa. Right underneath these, let's do this one in green. Right underneath them is going to be these other receptors here. These receptors are just beneath the actual epithelial layer. They're subepithelial tissue right here. These are actually responding to certain types of debris, harmful chemicals, certain types of matter that could be floating through this area here. Certain things that could cause irritation. So certain types of irritants. We don't have to go through all of the irritants, but basically anything that could actually be like pollen or allergens or certain types of noxious fumes or noxious stimuli, whatever it might be, noxious meaning that it's harmful, right? They will stimulate these irritant receptors. When these irritant receptors are activated, guess what happens? They move along just with the actual vagus nerve. And then they go into same centers. They go into the actual respiratory centers. So again, what's happening here? Whenever there's certain types of irritants like noxious fumes or certain types of debris or pollen or chemicals that are irritating the mucosal layer, it'll activate these irritant receptors. When the irritant receptors are stimulated, they'll send impulses down cranial nerve 10, sensory afferent fibers, down through the vagus nerve. These impulses will come into the respiratory centers within the pons and the medulla. What will they do? Well, think about it. If you've ever had something irritating your throat, what are you going to do? You're going to cough. You're going to try to pop that sucker out, right? That's one thing. What if you have irritation maybe a little bit higher than the actual respiratory tract, a little bit higher? Because these can actually come up, a little bit higher too, not just within the bronchi. They can go up through certain parts of the conducting zone. So if there's stimulation up there, they might trigger a sneeze to push something out through the nasal cavity and for the lower structures. What else could they do? They can actually narrow the glottis. And it can also trigger what's called tachypnea also, okay, which is again the accelerated respiration. Okay? So in situations in irritant receptors, they'll send these signals down through the afferent fibers of the vagus nerve and do what to these respiratory centers within the medulla and the pons? It'll let them know to trigger a cough reflex or a sneeze reflex or tachypnea, which is accelerated respiration, or it can also trigger certain mechanisms, right, that are trying to be able to get that structure out, whatever it is that's causing the irritation. Okay? All right, so that's the irritant receptors. Okay, so let's do another one, which is in this area. That's going to be right here. Okay, so there's other receptors here. They're actually called a really weird name, Juxta, capillary receptors. And there's two types of fibers. There's actually some called J fibers and C fibers. And all these J fibers and C fibers are, is they're going to be kind of around the lung parenchyma, so the alveoli and the respiratory bronchioles, that's all the parenchyma is. It's the gas exchange sites. It's also going to be here within the interstitial fluid, so the structure between the pulmonary capillaries and the alveolar structures here. And it can be right in this area. And all these J fibers and these C fibers, they're responding to a lot of fluid. So they're responding to a lot of fluid accumulation. What do I mean? Okay, let's grab this nice blue colored marker. Let's say that you have pulmonary edema. You have pulmonary edema, maybe due to left-sided heart failure, and the blood is actually backing up and it's actually causing fluid to accumulate within the respiratory membrane area, right? Or you have pneumonia. So maybe you have pneumonia, right? So pulmonary edema, maybe due to left-side heart failure, or pneumonia, it's causing the fluid to accumulate within this across this respiratory membrane, thickening the respiratory membrane. What will that do? Okay, well, in situations in which these actual J fibers or C fibers are stimulated due to a fluid accumulation, like pulmonary edema or pneumonia, it'll activate these receptors. When these juxtacapillary receptors are stimulated, they can also send impulses to the respiratory centers within the medulla. Let's bring this guy through here. All right, and look what can happen. It can also come in and affect the respiratory centers. What is it going to do within the respiratory centers? Whenever there's this type of situation, it can actually trigger kind of like a very rapid and slow breathing process. Okay, so whenever someone has fluid accumulation, right, so they have pulmonary edema, they have pneumonia accumulating there, the fluid accumulates, they can have dyspnea, so it's actually shortness of breath, gasping for air. And what else can they have? They also will have these impulses come into the respiratory centers within the pons and the medulla and triggering a very rapid but shallow breathing process. So again, these will actually trigger a very rapid yet shallow breathing process. Okay. So again, the juxtacapillary receptors are stimulated by fluid accumulation like pulmonary edema, pneumonia, or left side congestive heart failure, right? Stimulated by within the lung parenchyma, which is the alveolar walls, or within the actual respiratory bronchioles, or within the interstitial fluid around the pulmonary capillaries. They're stimulated, they send impulses to the actual pons and medulla respiratory centers and trigger two things. One is it can actually trigger this rapid and shallow breathing process. And another thing is that actually will cause them, because of that actual thickening of the exchange membrane, they'll have dyspnea, right? They'll have a hard time being able to breathe in air. They'll have struggle, they'll have shortness of breath, so they're gasping for air. Okay, so what's this last one? So remember we have these skeletal muscles. Now, within the skeletal muscles, you can have the tendons. The tendons have these special types of proprioreceptors. The muscle spindles have proprioreceptors. You even have what's called Golgi tendon organs. There's proprioreceptors within the joint capsules. But basically, what are proprioreceptors? Well, proprioreceptors are basically, they are these receptors, which are again located within the joint capsules, the muscle spindles, the tendons. And basically, what they're doing is they're helping us to determine our position within space, right? So they're telling us the position of our muscles, our tendons, and our joints within an actual specific type of space. Now, whenever these proprioreceptors are activated due to movement, so let's say that I'm actually having movement. So I'm exercising, I'm going on a run, whatever. But whenever there's movement, you stimulate these proprioreceptors excessively. So whenever these proprioreceptors are stimulated, they're actually going to send this information into the actual central nervous system. Let's see how they do it. Let's come over here to the left side. Okay, we're going to cover this pathway in more depth in neuro, but let's say it's due to muscle spindles, so we'll put MS for muscle spindles, Golgi tendon organ, joint capsule receptors, whatever. We could keep going through these suckers, right? But again, the overall result is that all of these are actually a group of proprioreceptors. Proprioreceptors, which are telling us the position of our muscles, our joints, our tendons within a specific space or area. Whenever these proprioreceptors are stimulated due to movement or exercise, it'll send these signals into the through the dorsal root ganglion, where the first order neuron is. It'll then come into the posterior gray horn, and it'll move into the dorsal white column. When it moves into the dorsal white column, it'll come up, right? And it'll come up through what's called the fasciculus gracilis. There's actually what's called the fasciculus gracilis. There's another one here called the fasciculus cuneatus. And this is actually forming what's called the dorsal spinal thalamic tract. Now, whenever the fasciculus cuneatus and the fasciculus gracilis, which is coming up through the dorsal spinal thalamic tract, come up, they eventually get to a certain part of the medulla where they cross. And whenever they cross, they actually form this next structure. We're not going to show them crossing. I'm just going to write it up here. This structure here, after it crosses, is called the medial lemniscus. Okay, so that structure right there is called the medial lemniscus. What the medial lemniscus will do is it'll send these signals to the actual DRG. Oh, man. When it tells this DRG, oh man, there's movement. So there's movement within the proprioreceptors. If this actual DRG is stimulated due to these proprioreceptor activation, what will it do? It'll tell the actual VRG. When it tells the VRG, what will happen? Well, the DRG is stimulated, the VRG is stimulated, what will happen to the action potentials? They'll increase. If the action potentials increase, what's going to happen to the frequency of these actual action potentials going down the phrenic and the intercostal nerve? It'll increase. If that increases, what happens? What's going to stimulate the external intercostals to contract and pull the ribs up more frequently and the sternum out? It's going to cause the diaphragm to depress. You get the whole story. What's it going to do to the respiration rate and depth? It's going to increase it. What's it going to do to the ventilation? It's going to increase it. Okay? And this is actually going to cause this increase in ventilation. But this type of action is not a hyperventilation which changes our blood gas chemistry. This is the type of ventilation that actually causes an appropriate increase in the respiration rate and respiratory depth. This type of action due to this movement is actually a specific type of increase in breathing. It's called hyperpnea. Okay? Hyperpnea. And this is an increase in the actual respiration rate and respiratory depth in response to movement or exercise or even painful stimuli. Okay? And again, it'll send these signals where? And send it up through the actual dorsal root ganglion into the dorsal white column, up through the fasciculus gracilis and the fasciculus cuneatus, which is a part of your dorsal spinal thalamic tract, which goes up and crosses at the medulla through the internal arcuate fibers and goes up as the medial lemniscus and stimulates the DRG, which stimulates the VRG, which increases the frequency of action potentials and increases the respiration rate and respiratory depth, which is the increase in ventilation, which is called hyperpnea. Oh my goodness, we have one last thing to talk about. Okay, you guys have been a sport if you guys have stuck in this whole way. I can't say thank you enough. Okay, so now inside of the brain we have a couple different structures that we have to talk about. I'm going to highlight them right here. One is actually going to be the hypothalamus, and the other one is going to be the cortex. So the other one is going to be the cortex. Specific part of the cortex, though, is actually going to be in front of the central sulcus. So they call this the primary motor cortex. And there's actually another one right in front of it, which is called the premotor. I'll put P-R-E, motor cortex. Now, these ones are very important because they are also, oh, one more, limbic nuclei. I've got to put this one in there, limbic nuclei. The limbic nuclei, the cerebral cortex, and the hypothalamus also influence our actual respiration rate and respiratory depth. Very, very simple, I promise. Think about this. So the hypothalamus and the limbic nuclei, these are very, very interesting ones. Let me explain. They're not very hard to remember. So think about this. Let's say that you're doing the polar bear plunge. So you're doing the polar bear plunge. Holy crap. Polar bear plunge. So during the polar bear plunge, what happens? You jump inside of freezing cold water. When you jump inside of the freezing cold water, what does it do? It can stimulate specific types of thermoreceptors. One of those things it can do is it can activate your hypothalamus. I want you guys to tell me for a second, if you guys have ever jumped in really, really cold water, what happens? That's what happens, right? That's what you do. Don't even lie. I know you guys do that. So what's happening? You're stimulating the hypothalamus, and what is that doing? That's affecting these actual respiratory centers. And what is it doing? It's causing that that increase in inspiration, right? All right, well, let's think about something else. Let's say someone said something that you did not like, and you're really angry. You're upset at them. Okay, they stole your graham crackers. I don't know, something like that. They did something that upset you, and they made you really angry. Have you ever been so angry to where you're just like, you're really mad, right? What's happening? The limbic nuclei, specific types of limbic nuclei, are actually receiving that stimulus, right? That sensation of anger, that person stole your graham crackers and you want them back. And if you get really angry, you start doing that. That's changing your respiration rate and respiratory depth. So I want you guys to think about that. And if that's the case, what is it going to do? It can also affect your actual respiratory centers. It doesn't just have to be that example. I gave you that example as a specific type, but think about if you're actually really depressed or you're anxious or you have some type of actual emotions, because basically limbic nuclei is playing a role with your emotions, right? So it's basically specific types of emotions like anger or happiness or depression, anxiety, whatever it might be, they can also influence your breathing. Okay, so that should be pretty simple. So hypothalamus, it's specifically responding to, hypothalamic and the limbic nuclei are kind of connected together. Okay, but I wanted to give you the example of the polar bear plunge because that's where the thermal receptors are. And the limbic nuclei are really responding to your emotions, but that's also coupled with the hypothalamus. One last one, and that's to the cerebral cortex. This is volitional control. So again, what do you call this? They call this volitional or voluntary control of breathing. Think about it. If I decide to take air in, I can do it. I'm going to do it right now. I did that on my own. That was voluntary. Think about this also. Let's say I decide to actually hold my breath. All right, you put me in a timeout and I'm mad and I want to have a tantrum. I pretend I'm going to hold my breath as long as I possibly can. That's also an example of voluntary control of breathing. So what happens is these voluntary controls can actually send signals directly down to the C3 and C5 nerves, as well as the T1 to T11 nerves. So I want you guys to remember that this voluntary control can actually send signals and bypass these actual pons and medullary respiratory centers and can go straight down here, and it can actually stimulate the ventral gray horn and stimulate the actual action potentials to go out to the diaphragm and to go out to the external intercostal. So they can actually do that on their own. It's called the descending corticospinal tracts, right? And on the same thing, we can also voluntary control to hold our breath. So if we want to hold our breath, I want to hold my breath, what happens? You know you can't hold your breath forever, right? And that's why, you want to know why you can't hold your breath forever? Whenever you're holding your breath, what happens to the actual ventilation process? It's decreasing, right? You're not pushing any air out, and you're not bringing any air in. What's the main thing that's happening whenever you're holding your breath for a long period of time? If you're holding your breath for a long time, guess what happens to the CO2 levels within the blood? They go up because you're not ventilating properly. If you're not ventilating properly, what happens to the actual carbonic acid levels? They go up in the cerebrospinal fluid. What happens to the proton levels in the blood? They go up. What happens to the central chemoreceptors? They get stimulated. What happens to the DRG? It gets stimulated. It stimulates the VRG. What happens to the action potentials? They go down and stimulate the muscles to do what? Take air in, inspire. So whenever you're trying to hold your breath, obviously voluntary control is limited. Okay, so voluntary control is when your actual, specifically from the cerebral cortex, can have descending corticospinal tracts that bypass the medullary respiratory centers and directly activate the nerves that are controlling inspiration. But it is only limited. Think about trying to hold your breath. If you hold your breath, you can do it on your own, but you can only do it for so long because the CO2 levels within the actual blood increases the protons, activates the central and peripheral chemoreceptors. And again, how much is this accounting for? The CO2, 70%. 30% within the peripheral. And if it does that, activates the DRG, which activates the VRG, which sends signals down to trigger inspiration, and then you stop breathing on your own, right? We stop the actual process of holding your breath. All right, engineers, we covered a boatload of information in this video. If you guys stuck in there with me, I can't say thank you enough. I appreciate it, guys. If you guys enjoyed this video, please hit the like button. Leave some comments down in the comments section, and please subscribe. All right, engineers, as always, until next time.